So let's look at some data that shows how retrograde messaging affects the presynaptic cell's ability to release neurotransmitter. And so here's the experimental setup, right? They're going to stimulate this cell and record the change in current actually from this cell. Okay, so we're going to be looking at flow of current, and because this is an inhibitory interneuron, we think we're going to be looking at probably GABAergic current here. Okay, so when they stimulate the cell here, they're going to measure here, and what they see is that iPSC, an inhibitory postsynaptic current. This is downward, right? So we think that this is inward current carried by chloride ions. This is our control. Now what they're going to do here is they give a brief stimulus to the postsynaptic cell. So you have to imagine that they're going to stimulate the postsynaptic cell first and then repeat their experiment. Okay, and what happens, right, when they measure, so step 1, stimulate briefly. Step 2, stimulate here. Step 3, record again here. And we can see that the inhibitory inward current is much reduced. Okay, so what do we think is happening? We think that this initial stimulus of this cell, when we stimulate here, right, leads to the endocannabinoid release that binds to these receptors and limits the amount of GABA in the next transmission. So I should mention that, how does this happen? That reduced inhibitory transmission, unclear. The book doesn't suggest much. I don't think we know exactly how it's doing that, but these endocannabinoids are binding presynaptic receptors and reducing the amount of GABA. So in that way, you could almost think of the endocannabinoids, right, as excitatory, because this neuron here, right, the one that we're recording from, is more likely to generate an action potential when endocannabinoids are present, because the inhibitory interneuron can't have as much of an inhibitory effect. Okay, let's look at panel C. C is a bit trickier to interpret, I think, because the axis is quite different. Okay, so they've sort of flipped the axis on us, and now we're looking at percent amplitude. So 100% amplitude is basically the largest inhibitory current that you could see, and 0% or something near 0% would suggest that we're seeing the inhibition due to the endocannabinoids, right? So, for example, and the setup is the same in B. So they're going to give a control pulse here, right? And they see the iPSC, that's this one here, as 100% strength. Okay, and then they give another one, before they do their little thing here, and then so they're going to see the same 100% strength depolarization in the postsynaptic cell. Here, 100% depolarization in the postsynaptic cell. But now they're going to do what we saw before. They're going to actually give this cell a stimulus here, briefly, right? So that's depolarizing the post. Then they're going to stimulate here, right, the inhibitory interneuron, and then they're going to measure what the response is, right? And so you can see that this response goes all the way down to almost 0. There's almost no response. Why is that? Because we think that the endocannabinoids that are released due to this stimulus in 1 retrograde signal back to the inhibitory interneuron, right? And that reduces the amount of GABA release. So we see very low iPSC amplitude, right? That it's 5% or 10% of controls, right? And this lasts for a while, right? And you can see that if we go out after a minute or so, right, there's much, much less of a diminished effect. Then, we can say, is this really an endocannabinoid-mediated effect, right? Well, this drug, Rimonabant, I think, right? This drug, Rimonabant, right? I don't know how to say it, but I know what it does, right? I know that it's an antagonist of endocannabinoid CB receptors. Okay, so now we're going to keep our setup the same, but we're going to wash the drug over it, and we're going to block endocannabinoids. Okay, we're going to block they still may be released, but they won't be able to bind to the receptors. And what happens when that's done? We still see 100% of a current, right? We still see the same current here that we would typically see in controls. So what happens? We depolarize V-post. We release endocannabinoids, but we block their signal, right? So they're not able to have any effect. So that means that GABA is still released. That means that GABA is able to bind to this postsynaptic cell, and we see this large inhibitory inward current. Okay? It's somewhat complex, somewhat complicated. So let me know if you have questions on how this is working. Okay, let's talk about the last neurotransmitter of the chapter, right? Another non-traditional neurotransmitter. Go forward here. Come on. There we go. NO, nitric oxide. Nitric oxide is unique. It's actually a gas. All right, and it's made when the amino acid arginine is metabolized. This is an A, metabolized, right? And so here's your amino acid arginine, and you can see there's an enzyme, nitric oxide synthase, and we produce nitric oxide. We also get some other byproduct, but we don't got to worry about that so much. It's not a big deal for us. Okay. As a gas, one thing that's important to note is that it's freely permeable. Right? It can diffuse right across cell membranes. And so what this means is that it's going to target nearby cells and signal locally. All right, so you could imagine a neuron that has arginine in this particular enzyme, and when the enzyme is active, it's going to release nitric oxide, and it can cross cell membranes, and it can affect cells locally. How local? Well, tens of microns. All right, and if you think of the cell as typically 10 or 15 microns, you can affect a neighborhood of cells, right? So the idea is that this gas is useful in signaling to basically neighborhoods of cells. Right? So if we want to coordinate localized regions, so coordinated regions. Right? Because it escapes. What about within the cell? How does it work within the cell? Well, it activates enzymes that lead to other pathways, right? Pathways that we'll talk about. So you can see in this case, in this diagram, right, it's activating this enzyme that's embedded in the cell membrane called guanylyl cyclase. So it works via activation of other enzymes. All right, and now that what you can see here is guanylyl cyclase being turned on can take something that looks a lot like ATP, GTP, and turn it on, right? And so now we take GTP and convert it to something called cyclic GMP, which activates a kinase. And we'll talk a lot about these pathways next chapter. But kinases basically cause intracellular change, whether they change gene transcription or translation or insertion in membranes. Kinases go on to have many, many effects, okay? So we think that nitric oxide is involved in regulation of other synapses, right? So this isn't going to be ionotropic, right? It's not going to open up any ion channels. It's not even G-protein coupled, right? There's no G-protein coupled receptor in the membrane. That doesn't exist. But what we do think is that maybe this thing called this protein kinase G can go on, and maybe what this does is activate some other ion channel. Maybe it allows it to stay open longer. Maybe it causes it to, I don't know, open slower, something of that nature. Okay, so what we can say is that it regulates more conventional synapses. Conventional synapses. All right, so typical synapses, like glutamate probably is the classic example, that this can modulate glutamate release by either affecting how easily the cell gets to threshold through some inactivation of some other ion channel. Right, we'll talk about those pathways, but our good example here is how glutamate is released. Right, just like we saw how GABA release is affected by endocannabinoids, glutamate release is affected by whatever targets protein kinase G has is a consequence of nitric oxide being present. Okay, so where do we stand? Well, this is the end of chapter 6, so all of these different neurotransmitters, all the way back to acetylcholine. I'd like you to know something if we mention it about how it's synthesized. I'd like you to know something about how it's packaged into vesicles, how it's released. We talked about the receptors. We talked about how it's taken back up or maybe enzymatically degraded. We talked about how they're repackaged in vesicles. We talked about the effects that they mediate in some cases, right? And so to varying degrees, I think we have different information for different transmitters, but I think generally that's the type of information that I want you to know, right? So I could give you the example, and I could maybe ask on an exam, explain how GABA release is affected by cannabinoids, and you'd have to go through some of the data that we talked about. Or I could say, explain how dopamine is synthesized, how is it put into a vesicle, how is it released, what type of receptors does it bind, what are some of its effects, and then how is it removed. It's also a fair question to say, what are some of the pharmacologicals that affect it and why, right? So any of that type of information I think would be fair game. So maybe making a big chart would help, or some flashcards with this information. But this is the type of thing that I would expect, right? To expect you to know. And we're going to go on. We're going to start chapter 7 pretty soon, which is going to be about the receptors. I think particularly the G protein coupled receptors. We've talked a lot about the ionotropic receptors, the one that open in response to ion binding, right? We've talked a lot about that. But now we're going to talk about some of the signals and the signaling pathways that happen when, a G protein coupled receptor is activated, or maybe when guanylyl cyclase is activated.